look a little bit deeper at some of the eukaryotic cells and understand where some of these organelles came from, in particular the mitochondria and the chloroplasts. So, the endosymbiosis theory, or the inside cooperation theory, explains how chloroplasts and mitochondria ended up in eukaryotic cells. The idea is that chloroplasts and mitochondria were actually on their own as individual organisms before they came into contact with eukaryotic cells, and that a cell that processed energy likely came to live inside of a host cell. So something that was a cell-like precursor to chloroplasts and mitochondria survived on its own and then got taken in by a host cell and just kind of became part of the host. Over time, they became dependent on each other and could no longer separate. The chloroplasts and mitochondria were forced to live with the cells, and the cells were forced to rely on the chloroplasts and mitochondria. Eventually, a cell that performed photosynthesis evolved into a chloroplast. So that's the idea, is that something like a chloroplast came from an independent cell that knew how to photosynthesize, and then that photosynthetic cell got taken in by a bigger cell, who then it started using the photosynthetic cell's process. And that the mitochondrion came from a cell that processed sugar and oxygen to make energy, right? A very usable form of energy. So this is what we're basically seeing, that at some point there was a eukaryotic ancestor, a prokaryotic ancestor, and that somehow they got kind of mixed together. The larger cell here engulfed the smaller one, and they merged into one, and eventually that cell became the mitochondrion. That means that this mitochondrion probably dwindled down a little bit. It's not as robust or as much of an individual. It's not self-reliant anymore, and the cell, learning its ability to create ATP and useful molecules like that, became reliant on the mitochondrion as well. And that chloroplast probably had a similar process as well. So if we look at how did this kind of even happen, how do we end up with more organelles like the smooth and rough ER and the Golgi apparatus? That's the idea that invagination happened. And all invagination means is that part of the plasma membrane folded in on itself, and we created compartments inside of the cell, including the nucleus, the rough, the smooth ER, and the Golgi apparatus. So the idea is that organelles have been developed by either endosymbiosis or invagination, or a combination of both. And there's some evidence that supports this theory, because both chloroplasts and mitochondria are similar in size, and they reproduce the same way as prokaryotic cells we see around today, using a process called binary fission. They both have ribosomes that are similar to the ones we find in bacteria, and those ribosomes are smaller than we find in the rest of the eukaryotic cell. One of the really big pieces of evidence is they have their own DNA. So chloroplasts and mitochondria have DNA separate from the nucleus. So if you were to look inside a plant cell, you would actually find 3 sources of DNA. You would find the nuclear DNA found in the nucleus, the mitochondrial DNA found in the mitochondrion, and the chloroplast DNA found in the chloroplast. It's also circular, meaning it's more similar to the bacterial DNA than the eukaryotic DNA. They also have 2 membranes, right? So they have a double membrane system in place. One coming from the original cell, the small cell, and then one from the host that engulfed it.